in computer, professor in computer science at Stanford. And in the past, he also served as chief scientist at Pinterest. I'm sure many of you actually used that. And also he co-founded the machine learning startup recently, Homo.ai. Jure is a really pioneer in the field of graph neural networks. As we all know, you learn in ML classes, and many of you are actually using the libraries that his group has developed, PyGs. And one of the most widely used graph neural network library. And his research spans foundational theory and methods in large-scale graph-based ML methods, but also many other applications in social systems, recommender systems, and also biomedicine. And I think the impact of his work, both on the core kind of foundational method and also wide-ranging applications is truly remarkable. His method has been used to combat COVID-19 pandemic, and it has been incorporated into products in many different companies. Facebook, Pinterest, Uber, YouTube, you name it. And I just looked up his Google Scholar. He has a citation of 217,000 citations. So that's.
That was last week.
That's what we got. So in this paper, not only publishes all these high-flying ML conferences, but also in really top venues in scientific journals like Nature and Science. Originally from Slovenia, Jure completed his undergrad there, and before earning his PhD in machine learning here at CMU in 2008 with Crystal Colusa. Unfortunately, Crystal has class today, so he cannot join us. And also, Jure did a postdoc with John Kleinberg at Cornell before joining Stanford as a faculty. So he has received many awards, and I will just name only a few. He was a Microsoft Research Faculty Fellow in 2011, and Sloan Fellowship in 2012, and Lagrange Prize in 2015, and also an ICDM, the Data Mining Conference Research Contribution Award in 2019. But I think beyond these, awards and all his accomplishments, his career so far has been truly inspiring. I think it's like from someone like me, as I've known Jure for quite many years and following his work. I think he continued reinventing himself. Right? It's not just kind of satisfied with one thing in one application, one domain, but he really tried to kind of continuously expand the impact and show what data mining, machine learning can really achieve for our society. And finally, I have to mention that we are also very proud to know that Jure is receiving the CMU Alumni Achievement Award actually tomorrow, and that's the reason that he's in town. And then he says maybe you should give a talk for students in the SCS and also for this Pittsburgh community. And so congratulations, Jure. And then so please join me in welcoming.
Good. Thank you. Can I speak this way? People hear me all the way at the back? Yeah, great. So super glad and humbled to be back here. I was a PhD student here 2004 to 2008 in the dungeons of the Wing Hall. I started in what was called CALD, so Center for Automated Learning and Discovery, and during my PhD it got renamed into the Machine Learning Department. And I'm just super happy to be here. I'm glad to be reminded that, I'm back to Pittsburgh, where being tough is a good value versus, being spoiled by the California sun. So it's been great, and I'm really glad to see that the university is doing well. New buildings are being raised, excellent research is being done, and it's just great to be back. So this talk today has two parts. So it's two talks, because the way this talk is titled, I'll just tell you what I'm currently really excited about, okay? And I'll first tell you something, give you some stories about what I'm excited about in the field of AI for science, scientific discovery, and biology. And I'll tell you then in the second part what I'm also excited about in terms of AI foundation models for structured data, predictions, machine learning, and things like that. So there are kind of two parts to the talk, and let's see how we go through this. But the first part of this talk it will be all about how can we speed up the rate of discovery by using AI, right? And the claim is that kind of the way what the science is limited by, it's limited by the amount of folks people like me can train so that they can do good discoveries, right? Like the amount of data we are collecting, the machinery we have to be able to do experiments and collect the data is increasing faster than we have the supply of experts who are able to reason, understand this data, test hypotheses, and move the science forward. Right, so the goal for the first part of the talk is to say, how could I use AI to dramatically speed up discovery, to suggest novel hypotheses, validate those hypotheses, better understand the data, and make these discoveries faster and in a more efficient way. So there are two routes in this, let's say, AI for science, how you could go, and they are nicely kind of complementary and they nicely build on this on each other. So the first part I'll be talking to you about is this vision of what we call AI virtual cell and specialized foundation models or large pre-trained models on all kinds of biological data modalities that allow us to better capture and understand data collected in biology. And I'll talk about the UC, which is the universal cell embedding model that we built, and the one that we are just about to release called Pulsar, that is a patient level embedding model. And then I'll also talk about basically how we can use AI to simulate behavior of human scientists. And I'll talk about our generalist AI scientist called Biomni, and then some cool work on top of it to do automatic hypothesis generation and validation in a statistically rigorous sense. So that's what we'll talk about. One thing that I want to kind of get across is that biology kind of really spans huge scales, right? It goes from all the way from small molecules to entire cells, to tissues, to entire patients, right? You have like orders of magnitude in kind of physical scales. And what we want to do is we want to be able to train models that can learn in a self-supervised, unbiased way directly from the raw data. And the key capability that we wanna have is we wanna have this few-shot or zero-shot generalization, right? We wanna have a general model that is useful for many downstream tasks, as opposed to, let's say, this classical machine learning modeling where, you know, we collect the data, we hand label it, and then we train a specific model for a specific task. I wanna train models without any human labels, without any human bias on kind of large amounts of unlabeled data. And the way we envision being able to do that is we have published this perspective in Cell earlier this year, talking about how could you go about building essentially a digital twin of, let's say, of a human cell or collections of cells, and how could these different models work together across these different scales of biology, right? Everything from understanding molecules, understanding individual cells, all the way to understanding tissues and patients. And the point is that the amount of data and the characteristics of the data that you have at these different scales is quite different, right? So hoping that you can build a single network that you can supervise end to end, that understands the structure of proteins as well as the disease of the patient or the phenotype of the patient at that level is just impossible because of the characteristics and the amount of data that you have. So the way we propose you have to do this is that basically you are in some sense stacking together and pre-training these specialized neural networks at different scales of resolution, right? So we want to do self-supervised pre-trained models at the level of molecules. We want to then have models that take these learned representations of molecules, DNA, RNA, proteins, and so on, and aggregate them together with other data modalities, maybe images and so on, to now create a representation of the cell. And then when I'm, let's say, representing a tissue or an entire patient, I'm now representing each individual cell in that patient that is informed by, let's say, the gene expression level at each of the individual cells of that particular patient, right? And if we are able kind of to stack these things together, then we truly have these large-scale embeddings that are informed by everything downstream in a truly kind of patient personalized way. It becomes interesting because you need to then start thinking about behavior and dynamics, right? Data is temporal. We are aging. Cells go through different states and so on. So that becomes very interesting. And also there becomes this interesting question about what I'm going to talk next, which is the idea of this universal representation space, right? So if I'm able to put these models together, then I'm able to learn in a unsupervised, unlabeled way, this representation space of biology, of biological entities, right? And the cool thing now becomes that if I have this space, I can start reasoning inside this space, right? So I come with this notion of virtual instruments, right? So if I have this kind of highly valuable, super rich, super dense representations that I have that come out of these models, now I can start reasoning inside this embedding space, right? So a lot of tasks where we are building task-specific models today on the raw data, I can just start building on top of these embeddings. And the idea is that now these models will work better because the representation that they are learning over is already informed of biology, right? So what can I do? I can start comparing different cells and kind of having these universal atlases of multiple different cells from different organisms, different species, all embedded in the same space. So I can start kind of reasoning what cell is similar to what and have this kind of reference atlas. I can start thinking about spatial organization, spatial niches. I can start thinking about, let's say, novel state discovery of cells, again, all in this universal space. I can start thinking about different types of perturbations, maybe some kind of dynamics of how the cell state is moving inside this space, maybe with some optimal transport type methods. I can start predicting how the representation or the state of the cell is moving through the internal or intrinsic or extrinsic perturbations, let's say small molecules or something like that. And I can really think about this as giving me an ability to do in silico experimentation and kind of thinking of this as digital twins. Okay, so if I have this universal representation space, I can do a lot of super cool stuff on top of it. So what I'll show you now is our path towards this vision and how we are executing it. And as I said, if you want to build this, let's say, digital twin of a cell, you kind of need to go through these scales of resolution from understanding molecules that are in the cell, understanding the cell itself, and then how the cells organize either into a tissue or even into an entire patient, right? And the idea is that we are going to build these self-supervised large neural networks that where at one level we are encoding the information and then sending it to the next level, to the next level neural network all the way to the multicellular scale. So one example of this type of model that we have built spans the molecular scale and the cellular scale. And this model we call universal cell embeddings model because it can embed any cell from any organism into the same underlying universal space. Okay, so you don't need to tune it, you don't need to do anything. You can take any cell from any organism and it's able to do this. The reason why it's able to do this is because it's a specialized architecture built ground up for biology, right? So it's not just, you know, your favorite transformer from Hugging Face and then somehow you create a sequence out of your biological data and train and hope you got something. But actually we went much deeper and really reasoned about, you know, how gene expressions are not the same as natural language. What is the proper self-supervised objective function that you train this method over? You know, it's not the next token prediction. And really the goal is that you want to have this fixed universal embedding space. The way we achieve this is that our input are not only the cell expression, cell level gene expression, this kind of single cell RNA-seq gene expressions, but for every gene, we know the protein sequence that gene is coding for. And that protein sequence is encoded by the molecular foundation model, meaning the encoded by the ESM-like model. So now you can basically take proteins from cells from any organism, as long as you know what proteins are in that cell, you can now embed any cell. And we are able to train this in a fully unsupervised way, no human labeling needed, and so on. And one thing before I comment on this plot here is that when you do these single cell RNA-seq experiments, these batch effects, these experimental effects are so huge that if you would take the same tissue and give it to two different labs to do the wet lab experiment, and then you would embed those measurements. One sample would be here and another sample would be there, right? They're kind of, these batch effects are so big that everything just clusters based on who did the experiment, right? But what you see, for example, here is that we take cells from, I think here, three different organisms, I think a fly, a mouse, and a human, and we color it by cells by which of these organisms they're coming from. And what do you see is that this data is well integrated, right? So the model, through this self-supervised training, learned how to do batch correction, how to nicely integrate this data without ever being told what an organism is or what a particular tissue or cell type is. Okay. And the interesting thing is you can do this actually even for organisms that you've never seen during training, right? So these are brand new data sets from different organisms that were never part of the training data set. We never saw what a chicken cells from chicken and mole rat and macaque, but the model is able to embed them. And what I'm showing you here is I'm coloring the cells according to the cell type that is annotated by the humans. And you see that, again, this kind of cell type organization naturally emerges from this self-supervised training of the model, right? So basically, the model learns how to organize human cells, and it organizes them according to the cell type. And if you go deeper and see, does the model kind of do these positions capture the underlying biology as we understand it today? The answer is yes, right? So the beautiful thing is that kind of biology emerges out of this. So this now gets you to the level of cells, but you can keep going, right? So now you could say, I can take the cells in your organism. Maybe I take cells in your drop of blood. I can single cell RNA-seq them. I can connect them. I can connect genes to the proteins. And now I can create a patient level representation that is basically the aggregation of the individual cell embeddings that we find. So we have this work that we are about to release called Pulsar that allows you to do to have a multicellular foundation model that takes in a set of single cell RNA-seq profiles and gives you a donor level or a patient level embedding, right? And the key thing is that now these embeddings are not just kind of created out of the blue, but they are informed by the cellular embeddings, and the cellular embeddings are informed by the underlying embeddings or representations of the proteins. And we are able to train this, again, in a completely unsupervised and unlabeled way. So we don't need any human labels. And now that we obtain these representations of donors or of patients, we can start asking, is this what is this informative of? In terms of learning biology with this foundation model, right? We have the molecular level information captured by the ESM2 protein embeddings. Those embeddings get aggregated by the cellular level foundation model into representation of each cell. And then the multicellular level is basically employs a transformer model to kind of capture cell-cell coordination while preserving the single cell resolution. And what's the output? The output is now the embedding or the representation of an entire patient. Yes.
The role of the ESM embedding here, because it's already part of the UC.
Yes. All I'm saying is the UC is taking ESM, the Pulsar is taking the UC, right? So it's like hierarchical. Yeah. Okay. And what's interesting right now, if you just take these patients, their blood draws, and you embed them with this model, and you, let's say, color the embeddings based on the disease that the patient has. Again, this nice coherent clusters homogeneous in color, so homogeneous in the disease, appear, right? So internally in our lab, we're kind of joking that this is the Theranos 2.0. Right, that like from a single drop of blood, we can create a patient embedding, and now we can do a lot of things with that patient embedding. And hopefully, we don't follow Elizabeth Holmes to prison, but that's, that's separate, okay? So the cool thing is that basically this organization, right, emerges without us ever telling what a disease is and who has which disease and so on. We can also do a lot of different experiments to show how informative these patient level embeddings are. For example, we can predict plasma protein levels better than the state of the art, or better than, let's say, pseudo-bulk type methods people are using. We can predict the age of the patient, this kind of immune aging clock, as people like to call it, more accurately than prior methods, or let's say either bulk methods or anything else, right? Again, based just on this embedding that was trained in a completely unsupervised way, right? And basically this finishes, I would say, the first quarter or the first half of the talk, really talking about this kind of idea of connecting the scales of biology in a set of aligned neural network models, and allowing you to create, embeddings that are very faithful at the patient level, but informed all the way down to the individual proteins. The reason we want to build these types of advanced tools is to do new science. So I want to now talk about the second part of work we've built, which is about how do we simulate behavior of, let's say, human biologists or human scientists to help speed up discovery. And the point is, right, like we think that drug discovery is this great, almost like, work where you go and you're working on discovering new drugs. But in reality, it's like navigating over 47 different Excel spreadsheets and using 19 different legacy tools, and, doing some literature searching and talking to your colleagues and doing some molecule design and drafting some protocols and so on and so forth. And this takes very long time. And it's super painful, and maybe at the end, we have some potentially useful result. And the question is, how could we help this, right? How could we shorten this process that it doesn't take, months or years, but it is much faster, right? So can we build kind of an agentic workflow that will allow us to speed up or automate certain parts of this process? And when I say an agent, I just want to kind of define what do I mean by agent, so we are all on the same page, right? An agent is a piece of software that is taking actions in some environment, right? An agent needs an environment in which it operates. The environment, let's say, responds back to the agent. Agent gets that response and can plan the next action. Okay, so if you want to build a scientist agent, you need an environment for science. So what we have done is we have built a basically, the way you can think of it, it's a Docker container that has about 250 different bioinformatics tools installed and ready for the agent to be able to use. So what is in there is all kinds of bioinformatics tools, all kinds of databases, data sets, ontologies, all kinds of data lake files, different AI models, lab protocols, and different know-how templates, right? So we took a lot of time to basically curate this machine that has Python and a bunch of tools installed, UC, AlphaFold, and everything else that is there for the agent to use. The way the agent operates is that it gets a high-level query from the scientist, and then goes and curates a multi-step plan about how it is going to execute that task while using all the advanced tools that it has access to, right? So this agent goes far beyond this kind of typical, let's do web search and literature review type of agent, to really, it can take your data, it can analyze it, and it can do this autonomously for half an hour at a time if needed. The way the agent operates is that it's basically writing Python code. So at the end, all this agent is doing is writing a bunch of Python code. It's executing that Python code. It's looking at the results of the Python code and continuing writing more Python. So why do I say this? This is important because everything is auditable, right? At the end, you get kind of a complete log, a complete audit what the agent did, right? Like something I would sometimes like from my PhD student, but I can never get it, right? But here, it's truly there. Every single step, everything is documented. You can verify everything. You can stop it. You can talk to it. It can explain why is it doing something. You can change the course of analysis and so on. So it's really for the human to be in control. It's actually interesting how well this works, right? We've done a bunch of different benchmarking here for a number of different, let's say, tasks that you do in biology, right? From rare disease diagnosis to drug repurposing to microbiome disease taxa analysis to single cell RNA seq annotations, some gene prioritization, variant prioritization, GWAS type analysis, perturbation screen design, and things like that, right? And the way we arrived to all these benchmarks is that actually we wrote a separate agent who is reading bioarchive papers and looking at what kind of tools and methods people are using in those bioarchive methods. From that, we curated this environment and we made it available to the agent to use. So it means all these tools that are actually meant for humans to use, we kind of rewrote them, add interfaces to them, so they are now easy for the AI agent to use. Just I can show you a quick case study. This is with one of the labs at Stanford, where they wanted to do a cloning protocol design, something I know nothing about, right? But here's the prompt. So I have this plasmid CRISPR-V2 blast, and there is a sequence, and they say, give me a cloning protocol. The agent came up with this 7-step analysis, kind of 7-step data analysis protocol using different tools. Here is the cloning protocol. It generates the plasmid map as well. These guys went to try it, and it worked, right? And then when we talked to the student, he was like, it would take me about 3 years of training, installing and hopping across many tools. Here, the agent did it in about 10 minutes. And it was correct. Another case study I can give you is also, let's see. This one is from Mike Snyder lab at Stanford. And what they had is they are doing this multi-omics approach together with physical activity and food consumption and things like that. And they have, about 30 patients, continuous glucose monitoring, some physical activity data, and about 460 different Excel files in which all this data was. So what Minta did is she uploaded this data to the agent and said, analyze this data, give me some plausible hypothesis. The agent went on this 10-step data analysis protocol, created a bunch of graphs as an analysis as a result of this, and came up with analysis and observations that Minta judged were actually quite relevant. So, she was saying, it would take a bioinformatics expert as myself, several weeks working several hours per day. Here, the agent was able to accomplish this in about 35 minutes autonomously. Right. So we have actually built this agent and open sourced it. It's called biomni.stanford.edu. In the first 3 months, we have more than 10,000 people using it. We have about several thousand monthly active users. There are people who are using it every day for different tasks. We have automated a number of different scientific tasks, meaning the total number of queries that is being used on the platform is now approaching 100,000. So it's really actively being used by the community. In terms of people from more than 4,000 different organizations are using it. What is interesting is that we have connected this agent with a bunch of other tools and a bunch of other data through partnerships. So we have a partnership with Ginkgo Bioworks, and all their data sets are available through the agent. SCverse, we did a really good integration. PyLab robot, they actually have a physical wet lab that now Biomni agent is able to use, and it's kind of able to run proper wet lab experiments. EHR data is integrated in there, as well as more than 200 million scientific papers, right? And it's the agent who now can call these types of tools to be able to make inferences and analysis. So if you'd like to try it, go to biomni.stanford.edu. You can try it. It's also open source. You can run it by yourself. There are two parts to it, right? There is the environment, the machine with all the software installed, plus the LLM who is driving the machine. We have, right now, we internally are using Claude and then a farm of machines on AWS, because every query gets a fresh machine so that the agent is able to do the analysis. And then you can kind of download the results. You can always also upload your data, right? It's for data analysis. It's kind of for data analysis. You can upload your data and analyze it with the agent, and it's all auditable. You can verify everything and stop it at any time and correct it and so on. So this is what I showed you now is an agent that can basically execute, let's say, bioinformatics workflows. But the interesting or the higher level prize is, could I go towards automating science? And what is science is all about? It's about validating hypotheses, right? And maybe what we know how to do now is execute a single work stream. But the question is, could I do general hypothesis validation? So we have this paper called Popper. We published at ICML this year, which is the idea is that we have an agent that imitates the falsification process. So the idea is whenever you have a hypothesis, you wanna falsify it. And so it's not about proving the hypothesis, but it's finding a way to refute it. And hypothesis is true if you cannot refute it, right? And this was kind of developed by a philosopher of science called Karl Popper. So that's why this agent is called Popper, right? And if you think about this, validating hypothesis at scale is super hard, right? First is the space of all hypotheses is super massive. The second problem is challenge is that hypothesis testing is something that is where you have to be statistically rigorous, right? You cannot just hallucinate something and pretend everything is okay. So you really want to have a guarantee that you are avoiding false positives. You want to know kind of what's your false discovery rate. And then the last challenge is when people usually talk about hypothesis testing, they're more thinking of this kind of small mathematical hypothesis, from A follows B and so on. Here we want to validate free-form text hypothesis, right? Scientific hypothesis. So the question is, how do we do this rigorously at scale? So there are, I won't go too much into the details, but there are several things what we do here. So what we do is we take this mother hypothesis and the type 1 error rate parameter alpha. And we have first an agent who is designing falsification tests, right? So it would take the mother hypothesis, so the main hypothesis, and outputs the implied sub-hypothesis, basically the null and the alternative, as well as the experimental protocol design, right? And this agent requires a lot of reasoning, and there is also a critique there that makes sure that falsification test is really robust. Now that we have the plan, we have then another step, which is the Biomni that takes now the experimental protocol, runs the analysis, and comes back with a p-value. And then right now that this is kind of one step, you can keep iterating and aggregating these p-values. It turns out that you cannot be aggregating p-values because these experiments are not independent. So there is a concept of e-values, and e-value is defined as expected value under the null hypothesis is at most 1. So you can now be aggregating these e-values, and that allows you now to do proper statistically rigorous hypothesis generation. And there is nice theorems we have in the paper and so on. But the main idea is that you have an agent who's proposing falsification tests, another agent that is executing the falsification tests. gets these p-values, we convert them to e-values, and then we keep aggregating these e-values until we can refute or reject the mother hypothesis. And just to show you an example, here is the prompt, right? Is that this particular gene regulates production of IL-2, right? Some protein. Here are different falsification tests that the agent comes up with. And then here are the corresponding p-values after executing this falsification test. And each of these falsification tests, is a multi-step kind of data analysis procedure. And what is interesting is if you do this and run it and validate it and see what it's doing, it's basically performing at the level of a PhD level scientist, right? In terms of type 1 error estimate, it's accurate, its power, it's about the same as the power of a human scientist. If you look at what kind of experiments it's doing or what kind of statistical tests it's doing, there is large overlap. So it's kind of behaving in a similar way to a human scientist. But where the difference comes is that it's about 10 times faster, writes about 3 times more lines of code, and does about 2.5 times more experiments in this time, right? So basically, this allows you to now start thinking about doing large-scale hypothesis testing, or maybe you can think of it even for exploratory data analysis in a fully agentic way. Okay. The last thing I want to say here is to address this question, but could we kind of move towards, scientific or biological superintelligence? And we have some evidence that may actually be possible. And what I'm showing you here is what we were doing RL-based fine-tuning of small language models and see can they improve at these scientific tasks. Okay. And I have 2 small models. I have this is Quen3, a 32 billion parameter model. This is how well it operates on our benchmark. Here is the latest, greatest language models, Claude and GPT-5. And of course the small model is about twice worse than these best models that we have today. But with a bit of RL-based fine-tuning, you can actually double the performance of this small model. This is the base model. So this is also a 32 billion parameter model. And you can be better than the largest models that are out there, right? So what I'm trying to say with this is, I think there is a great promise to actually train these LLMs to do statistical reasoning, to do scientific reasoning, to teach them how to run experiments, and push this level of performance even higher. Okay, so to summarize this part, right, is like I think the future of AI is truly agentic. The future of science is agentic, and that in the future we are going to have agents, tools that are basically helping us have an impact of an entire specialized team, right? And I think this is very fundamental because there are not too many places in the world, kind of that are as fortunate as you guys here or we at Stanford, where you really have kind of a critical mass of expertise to do something amazing, right? You have computational people, you have biology people, you have medical people, all these people together working on something, right? But with these types of agents, you can kind of distribute this expertise to wherever in the world, so everyone has access to these resources. So research becomes much more universally accessible, and we are dramatically reducing barriers, right? The key here, I think, is that the goal is not to displace the human, but it's to provide a companion or a tool for the human scientist to use and to kind of amplify the human potential, right? That's how we've been doing this. Of course, there's many good questions about what kind of hypotheses these models are coming up with, how are they testing them, how reliable and robust this is, and so on. But maybe the good point here is that everything we are doing is open source, available for everyone to take, build on top, scrutinize, and kind of together make improvements in this area. So that's what I wanted to say about the first part. Let me now tell you about the second part. Okay, so this is now a completely different topic that I'm also excited about. So what I'm very excited about is how do we reason over structured data? Okay, so if you say every organization in the world, including this one here, right? We have tons of this structured data, right? We have student records, their grades, class attendances, and so on, right? If I'm a company maybe selling something, I have my catalog of clients, I have my product catalog, I have data about what each clients have bought, when have they paid back to me, and things like that. And what I like to use this data for is I use it for decision making, right? And the way I use it for decision making is to kind of forecast or predict the future, right? If you think, how are these models being used today? We interact with them tens of thousands of times without even realizing, right? Whenever I swipe a credit card, it goes through some fraud model that says, how likely is this transaction to be fraud? Whenever I visit any of the websites, YouTube, Google, DoorDash, Netflix, every movie, every video, every dish, every restaurant there is there for the purpose because somebody predicted that is relevant, that is interesting for me, right? If I go to a social media site, again, those posts are there, prioritized, personalized to me, so that they engage me, right? And the point of all this is that basically the reason we are building these predictive models over this type of structured enterprise data is because we want to be making decisions at scale, right? I'm kind of turning uncertainty into action, right? I decide, do I block this transaction? Do I manually review it? What do I do with it, right? I can do this at large scale, at millions predictions a second. I can decide which patient to discharge, estimate the let's say the risk of readmission of that patient. I can estimate, forecast the lifespan of a patient and things like that, right? So these all are predictive problems. As I'm collecting more data, I can learn from it and make those predictions better, right? And in the age of AI, what you could do is you could say, okay, I have my database, I have all these tables, I'll just JSONize them, put them in a big prompt, and I'll just go ask ChatGPT, right? So you could give your table in a text format or a set of tables to ChatGPT and ask it, estimate, should I can I discharge this patient from my hospital? Or you could ask it, is this transaction fraudulent, right? And if you do that, you will get really terrible results, right? The LLMs were not built to reason over structured tabular relational data. They are built to learn over sequences, and they're not really trained to forecast the future, to estimate risk and things like that, right? So this doesn't work. So what are you then stuck with is this kind of classical machine learning that a lot of people in industry are doing, right? You take your original data, you do some ETL data cleaning, you engineer the features, you create your training data set, you train your model. Now you want to put the model in production. For every of the features, you have to create a separate kind of Hadoop or Databricks or SQL type workflow that is computing those features. Those features might be stale, might be out of date, and your model performance degrades and so on. So when you look at this, you need about 2 employees per model in production, right? So it's like super expensive. It costs a lot. And many of these models are never put in production because doing this manually is so hard and so slow. And just maintaining these models is a total nightmare because they're constantly degrading in performance. Somebody has to be constantly adding new features to do something with them. So what my point here is, there is this great world of LLMs. I just give it my contract. I can ask it questions about it, and it just answers. It's like beautiful. But then if I want to do something on this structured side of the data, I'm stuck with machine learning, right? I'm stuck with feature engineering. It's slow. It's terrible. So what I want to show you here is to talk about purpose-built foundation models that are meant for predictive tasks on structured data. So they are meant for estimating how likely is this transaction to be fraud? How likely am I to run out of stock for this particular product next quarter? How likely is this lead becoming to convert and become my client let's say in the next 30 days. Okay, so all kind of machine learning prediction problems. And the idea is the following, right? The idea is that we will connect to the raw data, whatever it is, even a lot of biological data you can represent in this tabular form, add all kind of external ontologies and databases to it to connect it, and it's super cool. And then we'll have one universal foundation model that allows me to make predictions. So the way this needs to work, it needs to be able to ingest any kind of data, any kind of schema, be it e-commerce, finance, healthcare, doesn't really matter. So that's the first thing. The second thing we need to be able to do, we need to be able to ask this model questions, right? And if you look at these questions, these are all predictive, forward-looking questions. And then this large pre-trained model that is frozen, right? There's no training involved, needs to be able to give me answers to these questions, needs to be able to tell me how accurate these predictions are, and also needs to explain why a given prediction is being made. Okay, so that's what we are trying to achieve. And what I want to just tell you, right, is as we all accept that, you know, we need special purpose-built foundation models for biology, we also need special purpose-built models for structured data. And, you know, in the same way as LLMs kind of take away tedious NLP work, RFMs take away this time-consuming, difficult predictive modeling work, right? So these are two complementary technologies that can really nicely coexist together and kind of complement each other, right? LLM is great at human level type reasoning. RFMs are great at prediction, forecasting, risk scoring, things like that over structured data. So let me tell you how this works and how you build this, right? The fundamental difference is that in classical modeling, you train your model and then, you know, you give it some data and it makes a prediction, right? So you have this training phase and then you have this inference phase. The idea in our case is that the model will be pre-trained, but we are not going to tune it or train it for a given data set or for a given prediction task. So the model will get access to the data, will get a prompt about what prediction task we are solving, and this frozen architecture needs to produce an answer. And you know, what's the insight? How do you make this work? The insight is that basically your database, your collection of tables, can naturally be represented as a graph, and you can then attend over this graph to learn from it. So I can take any collection of tables. I'll be using this example of users, products, orders, and let's say website clicks. I can represent this as a graph, and then I can train a graph transformer type architecture. So it's like a transformer, but transformer attends over a sequence, but this kind of transformer attends over the underlying subgraph to give me a prediction. Okay, so a bit more details. The idea is that for any schema, I can represent the underlying data as a graph. Every table defines a node type. Every row defines a node. The corresponding columns define attributes of these nodes. So here I would have user nodes connected to their purchases, connected to the products they purchase. And imagine now I want to make a prediction whether, let's say, this user is going to churn. What I'm going to do is I'm going to go and I'm going to attend over this subgraph centered at this node, right? So the attention mechanism is going to attend over the raw events of this person, is going to attend over the products that these events are attached to, is going to attend over other events of these other people who have some more events on these other products and so on, right? And through this recursive notion of attention, I can basically attend directly over the raw data without needing to do any human-based feature engineering, right? Because feature engineering is just kind of creating an arbitrary summary over the data, right? But here, I'm not creating that summary. I'm asking the attention mechanism to attend over the raw data and create that summary for me. And of course, this is a bit more complicated than classical transformers because you're not attending over a sequence, but you are attending over this recursive graph structure. So your notion of positional encodings and your notion of the attention mechanism gets quite different. But the benefit is that now you can build much more accurate models. The reason for that is that the attention mechanism has kind of more fidelity than any human hand-engineered feature. So it means that you can now learn directly from the raw data to learn correlation, filters, aggregations, whatever the attention mechanism learns that is predictive of the downstream task. And as I'm telling you this, this is like supernatural, right? We've seen two instantiations of this already, in the last few years, right? If you think about old school computer vision, we took the pixels and we did some feature engineering to detect, Gabor filters and SIFT features and so on. And then we would train a classifier on top of that those features. And of course, it didn't work because those features we extracted from pixels were imperfect, right? So what do we do today? We just have a neural network and say, here's a set of pixels. You learn how to transform pixels into a prediction, I don't know, whether there's a human on the image or not, right? Similar, I think, revolution happened in natural language understanding, right? We used to do all this kind of parsing and entity resolution and all that. Nobody's doing that today anymore. You just have a sequence of tokens and you push them through the neural net. And what I'm doing here is exactly the same. The only difference is that I'm doing this on a database. So I'm representing this as a graph and pushing that through the neural net. And what the effect is? The effect is that I can now build predictive models that have superhuman performance. I can build models that are better than any human data scientist can build. And not only that, you can build them much faster because you completely sidestep the feature engineering step, right? You just learn them directly on the raw data. Okay. And that's quite exciting because now this means you can put these relational deep learning type models in production very quickly for a number of different predictive tasks. Okay, but I want to show you that you can do even more. And what you can do is you can actually build a large pre-trained model that can apply to any database and to any predictive task. Okay. And if you think about this sounds crazy, right? Because you say any schema, any set of tables, any organization of tables, any number of columns, any type of column, any organization of column types, I would just like to learn over that and make a prediction. So the way this works is the following. So I'll explain this and then we'll go step by step. So the first thing I need to connect to the database that's on the top. Then I need a way to prompt the model. So I have this predict domain-specific language. I'll talk about it. Then from that domain-specific language, we are going to generate a set of in-context examples. We are also giving the model one kind of unlabeled example. These are my subgraphs from previous slides. And in a single forward pass of this model, you are getting an accurate prediction. The question is, how does the prompting work, right? For LLMs, we prompt them with natural language. For these relational foundation models, we prompt them in this domain-specific DSL. They operate on top of a database. They do in-context, relational in-context reasoning to give you an accurate prediction. So the way this works is that we have developed a domain-specific language that has kind of two parts. It has the predict part and the for each part. And predict defines what is the target we are predicting. And then we are saying, who are we predicting this target for? And if you just read this, the first one says, predict what is the type of the type of the first order that you are going to make in the next 7 days. The second one says, predict me the count of orders 7 days in the future, is that greater than 0? So that's a binary classification type. This one is saying, tell me the sum of order values, let's say 7 days in the future for a given user, right? So that's modeling how much a user or a customer is going to spend in some future time period. And this one is a recommender system. It says, predict me the least distinct orders of item ID, again, let's say 7 days in the future for a set of users, right? So the underlying model, we prompt it this way. We basically say, what's the quantity we want to predict for what entity? Okay, so now that we know what we can predict, and because the data is temporal, right? Events come in over time, I can basically do time travel and pretend that, this is the time today. So I can sample a proper subgraph around that entity, but I can then look 7 days in the future to see what's the count of transactions of that person, or what's the sum of transaction values 7 days in the future. And this way, I can generate a small, you can think of it as a training data set, plus at the end, I give it an example of the user I'm interested in at the present time, and put that into my foundation model so it makes a prediction, right? So I'm creating a task specification in this DSL that generates a set of in-context training examples, up to 10,000 in practice. I have then a set of unlabeled examples for which I would like to make that prediction, and a single forward pass of this relational graph transformer architecture that is pre-trained and has never seen this data is then able to give you that prediction. And I'll just show you how this works. Here's a database. Here I'm typing in this query that says predict me whether the count of orders next 30 days is greater than 0. And I want to predict this for some given user. And then here we are generating in-context examples. Here's the prediction, probability of false, probability of true, and a natural language-based explanation, right? Without any model training, without any model updates, you can basically do in-context machine learning. You can also ask yourself how accurate this is, and we have tested this with PhD level data scientists, all kinds of machine learning methods, graph neural networks, as well as LLMs. And across entity classification, for example, what we see is that a pre-trained model is essentially as good as manually built specialized task-specific models. This is for classification. This is for regression. Again, you see very close here, but this one is much better because this is error, so lower is better. This is for recommendation. And the last one I'll show, this is actually the benchmark coming out of SAP. This is business data. It's some supply chain management, some sales office prediction task, plant manufacturing prediction tasks. And again, you see that, let's say, these AutoML methods are about, let's say, 0.79, while the foundation model is at the same level. If you fine tune, you get to the superhuman regime in terms of accuracy. So I think this is very exciting because it kind of shows how we can make machine learning much more interesting, right? We can basically have a pre-trained model for machine learning that gives us the same accuracy on a novel data set and on a novel predictive task as manually built models. If you want to fine-tune this, you can push it even higher. So to conclude, the part of this talk, what's the insight? The insight is that you can take any database, represent it in this graphical form. You can generalize the transformer architecture to be able to attend over this graph. And now you have basically a large pre-trained model that can do reasoning and prediction directly over this data. Why is this, I think, interesting is because it democratizes predictions, right? It allows it kind of the focus goes from tuning neural network architectures to really thinking about what data do we want to use, how do we want to curate that data to get out the predictions we want. So this is the last slide I want to show. So what I think is surprising here is that you can actually have a pre-trained model for machine learning tasks that is able to do in-context learning across any data set, any database, multi-tabular. That's, I think, crucial, not single table. And that by fine-tuning it for a specific task, you basically get to this superhuman accuracy regime. The benefit is no feature engineering needed. Let the attention mechanism attend over the raw events, and this way extract much more signal from the data than manually designed features or data summaries are able to do. So this is what I wanted to show. Just wanted to acknowledge a number of people from my lab, collaborators, more collaborators on our Biomni project, and very happy to take questions.
To ask questions, we have the room until we have time, so especially the students, feel free to ask questions.
Sorry to run a bit long.
ask a question. So this is related to your Biomni project. In healthcare, it's really important for models to be interpretable. And if you have these embeddings, you lose that sort of interpretability. Have you addressed this question in your work?
Good. So the question is about interpretability. I'm not sure why would you say embeddings are not interpretable. I think these models are very interpretable because even when you create an embedding, you can run the embedding model backwards and ask, what did you attend over? Right? So what is nice is that basically it tells you exactly what raw data, what raw signals it attended over to create that embedding that you are then using for whatever downstream task you are doing.
Maybe a better way to ask the question, you had the example with human bee and rat data, if I remember correctly. There were a bunch of patches. Is that like qualitatively interpretable? Like
what those patches were?
Yeah, exactly.
Yeah, if you would look at those patches, those would be different cell types. Okay. Right? So I know the next slide had all these different colors. There the patches were colored by the cell type that people annotated these cells with. The model never knew what a cell type is, was never optimized to create these clusters of these patches, but it was very nicely homogeneous, which basically says that the model in an unsupervised way was able to discover cell types. Yeah, thanks for the question.
So I wanted to ask you about one word I saw in one of your slides that you didn't say anything about it. So in when you're talking about Pulsar, I saw the word guardrails. I'm very interested, what guardrails are you thinking about putting in for designing experimental workflows, and how are you doing that?
Good question. Especially at the Biomni level, I would say there are all these, let's call them biosafety protocols and things like that. And the way we are thinking of integrating that right now is having basically a judge who is kind of overlooking, being prompted to decide what can be done and what is, let's say, unethical, or what questions shouldn't be asked. So that's how we are thinking about that. We are also creating kind of red teams who are trying to break the system that gives us training data so that we can then actually train the guard even better. Those are some of the things we are doing. Thanks for the question. Yeah.
I was curious about the Biomni project specifically, where you were assessing the performance of the model versus scientists. You talked about like the frequency of amount of hypotheses that were generated, as well as lines of code. But I think an interesting metric is like, are these lines of code productive and useful? And also how do you go about like verifying these hypotheses? So could you talk a little more about sort of that?
Yeah. Great question. So when I had, I'll just put it up. You were asking here, right? So in terms of is this valid, there are the first two plots, right? You are about type 1 error, right? Like you basically say how accurate. What is your false positive rate? That's the type 1 error, and you see that copper actually has lower false positive rate than humans. And then this is in terms of power, which is what is the accuracy when you are falsifying this hypothesis, and it's comparable to the human. The way we've done this here is that we designed benchmark where we knew whether the hypothesis is true or false, but then we would permute, or we would take, let's say, statements that are true, but we would permute the data, so we knew then the statement is false. And then we were basically able to, we said, can I prove that this statement is false? That was the experimental protocol we were doing here, right? So we would take true statements that are based on the data, we would then permute the data, let's say permute gene expression levels, so that now we know that the outcome the data does not support the outcome. The question is, can the agent prove? So those were some of the tests we were running to understand how good this is doing and how reliable it is. There are failure cases, of course, and one of the failure cases we noticed is that when the falsification test has nothing to do with the mother hypothesis, right? You could say, you can have some biological hypothesis, but then the falsification test is, does the water freeze at 0 degrees centigrade? And you can prove that or disprove that thing, but that p-value is not informative of whatever the mother hypothesis is. So we see, for example, sometimes that these falsification tests that the model comes up with have nothing to do with the top hypothesis. So to do this, we require actually large reasoning-based models where we see that they are able to come up with good falsification tests. But that's one of the examples of failures. Another type of examples of failure sometimes happens that just, the model wrongly reads the p-value. It does everything correctly, but then you ask it, okay, now extract the p-value from this experiment. There's some printf p-value, and the model reads it wrongly. Like, it's silly things like this that also happen sometimes.
I wanted to ask about these relational foundation models. First, if you're data set agnostic and the forward pass is for prediction, but ultimately the inference is relational and just a graph over a data set, what is the universal approximation, or like what is the sort of universal representation space of that embedding model? I can see it for UCE, it's quite intuitive because it's on the basis of an individual cell.
Yeah.
But you can even imagine that the problem posed by UCE is this for the pulsar kind of at a higher level. It's just a subset of the problems that the relational foundation model could resolve.
That's a great question. So there are several hard problems. Maybe for the relational model, there are two hard problems you need to solve. The first hard problem you need to solve, you need to be basically able to universally encode every row, right? But some rows can have lots of columns, some rows can have few columns, some rows can have time, can have, categorical variables, strings, images, whatever is in there. So that's the first thing you need to teach the model is how does it basically encode this row in some kind of universal, almost like domain agnostic way. That's the first part. And then the second part that you need to teach the model, you need to teach the model the in-context learning, right? You need to be able to teach it how to do at inference time, learn from the in-context examples and generalize to that unlabeled example, right? So when we generate training data for it, we use a lot of data augmentation and a lot of almost like synthetic machine learning models that then are used to teach the transformer model how to spot patterns in the data and how to generalize them to the layer. That's what I can say about it. The last thing I would say is actually these models are not that big. They're sub-billion parameter models, similar like 20 million parameter models as well. 100 is already awesome. But it's much smaller than LLMs. Just want to say that. Yes, question. Go ahead.
Of Biomni, since Biomni is a general purpose agent can address many tasks, but there's also like specialized agent in biomedical domain, like CRISPR GP for CRISPR, spatial agent for spatial tasks. So I'm wondering how Biomni compares with those specialized agents and what's their relationship? Will Biomni eventually including those external design agent?
Yeah, great question, right? So the way we are for Biomni, we wanted to do something general. But of course, the agent is only able to do as much as it has access to the data or the tools. What we have also noticed is that these specialized agents, what they have is they basically have some specialized knowledge, either in some kind of task templates and things like that. So in Biomni, we can easily ingest all that data, so it's kind of so that the agent is able to use it. So what we have seen now through open source is that a lot of people are contributing their own tools, as well as their own kind of knowledge bases, task templates, and things like that, so that the agent can use them and perform better. And I mean, to me, just kind of to say, I think there are two questions here, right? I think, even if you don't believe in AI, just having something that can execute your bioinformatics workflow without you fighting Python versions, downloading random GitHub repositories and spending a day making sure that the versions matches and everything, even that is already immensely useful. And whatever the AI can do on top, that's just a cherry on the cake.
Thank you very much. So real quick, for the relational foundation model, I'm just curious, did you notice any particular like scaling laws with respect to the data or the model size that you thought were interesting? I mean, I imagine it scales differently and excitingly.
Great question. So scale, like it's interesting, right? Like these models depend kind of much less on the semantics and this kind of general knowledge that you want to push into the LLMs, right? So these models are kind of much more about understanding patterns and things like that. So you get two types of scaling. You get the scaling as you make the model more powerful, that it can kind of reason better. But then you also get, in some sense, scaling with the amount of training data or in context examples that you put in, because the more of that you have, the more accurate the predictions. Yeah, thank you.
Can I take one last one? Yeah.
So question about Pulsar's application on utilizing it over cross-sectional data foundations. Like could you use it to predict cellular phenotypic changes over time?
Can you repeat? Sorry.
Sorry. So it's a question about Pulsar's application over cross-sectional data foundations.
Yeah.
Could you use it to incorporate the dynamicity of cellular phenotypic changes over time?
So the way I think the answer is, I don't see why not. Okay, so perhaps yes. So the way the model, like what the model does, it takes a single cell RNA-seq data of a drop of blood and gives you an embedding. So now if you draw blood at different points in time, I can now create time-resolved, let's say, embedding or a trajectory of a given patient. So you can use it for that. You could also be using to say, let me embed a patient, and now maybe in this embedding space, let me create a virtual instrument that is predicting where the patient is evolving, where the patient is going. Or you could use some kind of optimal transport method that would try to infer that trajectory. All this is possible. What we are using it right now for is for some CAR T-cell therapy and understanding when can we cure cancer and when not, for example. And it seems to be working well.
Yeah, so just like a very high level question. So like you presented a lot of like cool stuff about scientific agents and things like that. And it kind of sounds like everything's perfect. So I just want to ask, like, what's like the next big thing, or like what's the next big block or like the next big development for scientific agents and things like that? Like, what's the thing that's just like stopping it from like blowing up? And
yeah, I mean, I don't think it's perfect. It's better, but it's not perfect. What the next things are? I think the next things are really in collecting reasoning traces of human scientists and further tuning or training these models on that. I think they are still quite unreliable. But for example, now with Biomni, we have 100,000 of these traces, and in the end, scientists are giving us feedback and so on, right? So I think from those trajectories, there is a lot to learn. Right. That's how we are thinking about it. Another thing I think that you can think about is right now you have this LLM and a set of tools, but in the future, I think you might be doing much more joint training of the tool and the agent at the same time, so that the agent and the tool are not separated by the Python code that the agent writes, but they actually communicate through tokens directly. Right. That would be something that would be super cool as well.
So before we, last question.
It's a high level question. So like you've had a really successful and impactful career. So I'd like to know, what advice would you give to a graduate student who wants to have a similarly successful and impactful career, especially since it seems like most of the traditional value is about to be replaced by the models you're writing?
Okay. I don't know if I have any, I can just say like how do I think? I'm a person who really gets bored very quickly, and I don't like boring stuff. So I only do stuff that is interesting and cool to me. That's why I think I also like to move around a lot, because I'm trying to do things that I'm actually really interested and passionate about. And even like in certain areas, like I did a lot of social network analysis and information propagation and social media and so on. And at some point in time, you kind of feel I said here what I had to say, and you kind of naturally move on. So maybe that's one thing. The second thing I feel like is I feel in a lot of research, we take too little time to think what is the good problem to solve. Right? We just like to be busy, and we like to do some next thing so that we can go into the mechanics of research. So we spend a lot of time in my group just thinking about what's the problem that's worth our time. Right? Like, will this be interesting, important enough? Will people care enough that this is worth my time? Right? And I usually ask myself, when I try to determine that, I ask myself 3 questions. I ask myself, why now? Like, why today I should be doing this research? Why not 2 years from now, or not? Why not 2 years ago? Right? So there has to be something interesting in the world that makes this research most valuable right now. Okay? And maybe this means, oh, I just got some exciting new data set. Oh, maybe there is a new experimental method being developed and the data is coming in. Whatever it is, maybe there is this interesting technical development that now allows for asking a new question. So now we can ask, right? So that's the first. Then the second one for me is, why should I be doing it? I have a lot of cool ideas, but maybe I'm not the best person in the world to work on them. So I try to be kind of honest to myself or to ourselves. Why should we be working on this, and why are we the best people in the world to do that? And then I try to ask, who will care? If I do this, who is my audience? Right? If I write this paper, who is actually going to read it? Who is that audience? And what am I trying to tell? Right? Because it's not just, oh, we did this research, it's cool, but it's like, who is this meant for? Like what kind of Google queries will people use to find it, right? Or who is expected to read it? Are these domain scientists? Are these computational scientists? Is this a mixture? How big is that community, and what does that community care about? So those have been some questions that have helped me let's say make progress in research. But I think the most important thing is, don't let anyone tell you no. Just do what you think is right. Ask yourself, what do you think is this right thing you should be doing, and that it's worth your time and your life, and the rest, I think, will work itself out.
Great. On that note,
Thank you.
Thank you again for coming.